Hello, hello, hello. It's just now 7 p.m. on Wednesday, November the 1st. And I want to welcome you all again to the fourth annual UCSB Natural Reserve System Fall Virtual Seminar Series. My name is Andrew Brooks, and I will be your moderator for tonight's presentations. I want to encourage everyone to join us each Wednesday night at 7 p.m. tonight and for the next 2 weeks as we virtually visit one of the UCSB natural reserves and showcase some of the truly incredible research and other activities taking place there. Before I kick off tonight's program, I would like to briefly go through a bit of housekeeping. The presentation portion of tonight's seminar will last approximately 45 to 50 minutes. We have set aside the remaining 10 to 15 minutes at the end of the presentations for any questions that you all might have for tonight's presenters. If you look at the bottom of your screen, you should see a little button labeled Q&A. You can click on that button and then type your questions into the text window. I'll be monitoring the questions during tonight's presentations, so please feel free to type your questions in at any time. Okay, with all that out of the way, I would like to begin tonight by introducing Dr. Carol Blanchette, the director of the Valentine Eastern Sierra Reserves. Carol will present a brief history of the two reserves, the Sierra Nevada Aquatic Research Laboratory and Valentine Camp, that together make up the Valentine Eastern Sierra Reserves. And then she'll discuss some of the exciting work that is taking place there. Carol will then introduce Dr. Chris Wall, our featured speaker for the evening. So with that, take it away, Carol.
Okay, thanks, Andy. I'm just going to share my screen here. And let me know if you can see that. Okay, welcome everybody. Really nice to be here with you all. Today, I just want to introduce our two sites that are part of the UC Reserve System. As many of you know, there's 41 sites now that are part of the University of California Natural Reserve System, and 7 of those are managed by UCSB. Today, we're going to visit 2 of the sites that are furthest from UCSB, and they lie at the heart of the Central Sierra Nevada Mountains. Both of these are high elevation sites between 7,500 and 9,000 feet in elevation. And they're in this region of the Eastern Sierra known as Payahunadu by the Northern Paiute people. Both of these sites are located within the ancestral territory of several indigenous people, including the Numu, the Nim, the Kizzetika, and they were seasonally used by these tribes as well as the Miwok and Shoshone people. So the first site we're going to visit today is Valentine Camp Reserve. And Valentine Camp is one of the older sites in the system. It's comprised of 155 acres, and it's been protected from grazing and entry since the early 1900s. So it has some really remarkably pristine features, including subalpine habitat, including montane forest, chaparral, Great Basin sagebrush, high montane riparian vegetation, wet montane meadow, and seep and spring vegetation. For those of you who either live near the Eastern Sierra region or visit here, this fall was a spectacular year for the aspens and the fall color. Valentine Camp also sits in an area that we refer to as the wildland-urban interface. So it's bordered on one side by thousands of acres of Forest Service and public land, including Mammoth Mountain ski area shown here, and bordered on the other side by residential homes and resort communities of Mammoth Lake. So Valentine Camp is just off to the left in this photo. The Valentine Camp property was purchased around 1916 by a group of businessmen from Los Angeles, and it was used as a hunting and fishing camp. So shown here are 4 of the original owners of Valentine Camp, Joe Cook, Joe Sartori, Henry O'Melveny, and then on the right is William Valentine, for whom Valentine Camp is named. Housing at Valentine utilizes the original log cabins that were built in the early 1900s. This is the cook cabin, which is still very much in use today by researchers and students. So you can see the large fireplace and dining area in the front here. And as I mentioned, Valentine Camp is known for, or named after, the Valentine family. On the right here is Mrs. Carol Valentine. She was a great patron of the arts and had a love for botany and horticulture. She was really instrumental in the development of lotus land in Santa Barbara, and she understood the botanical significance of Valentine Camp. So back in 1972, she approached Vernon Cheadle, who was then the chancellor of UCSB at the time, also a botanist, with the idea of donating Valentine Camp to become part of this newly formed UC system of natural reserves. And so Valentine Camp officially became part of the UC NRS in 1972. And according to Mrs. Valentine, donating Valentine Camp to the UC NRS was the best possible way to preserve the land and use it beneficially. So Valentine Camp today is still used by a wide variety of researchers studying topics ranging from plant biology and forest ecology to wildlife. It's also a site that is used by many university research classes in fields ranging from biology to geology. And the location of Valentine Camp sits just below Mammoth Pass, which ensures that it gets a huge amount of snow each winter. And given the rustic nature and the uninsulated nature of these cabins that are fully buried under snow each winter, we only operate this site for use by researchers and classes in the summer months. So this is our education center building at Valentine. If you hadn't heard, or if you were part of coming up to Mammoth at some point in this last winter, we did get a great deal of snow and we were pretty fully buried. This is the education center in March. And this is that same building just a few weeks ago, just to give you a sense for how much snow this site receives in an average winter, and a lot more this past winter. One of the signature programs we have at Valentine Camp is an outreach program where we offer public tours throughout the summer months. So every week we have several tours on topics ranging from history to geology, maybe bears, wildflowers, and more. The tours are typically around 3 hours, and they provide an opportunity for people to come hike around the reserve's 155-acre property and then see and hear about a wide range of different habitats and research that's happening there. Another large part of what we do at Valentine Camp is our outdoor science education program. And part of this program, we host over 2,000 school children each year from schools all across Mono and Inyo counties. So these are primarily elementary school children from grades K to 6 that come to the reserve, both Snarl and Valentine, each year for grade-specific field trips, lessons, and activities. And then in the summer, we also offer week-long camps for children on a variety of different topics. So the other site I'm going to take you to that's located about 15 miles away from Valentine Camp, just outside of the town of Mammoth Lakes, is the Sierra Nevada Aquatic Research Lab, better known as SNARL. And SNARL started out as a U.S. Fisheries Bureau research facility in the 1930s. And so here is SNARL back in the 1930s. The station then was referred to as the Convict Creek Experiment Station, and the focus of the work was the emerging trout fishery. It really focused on the behavior and survival of these introduced trout in natural streams. And ironically, today, much of the research at SNARL still focuses on the ecosystem impacts of these trout introductions and the consequences for many of the native Sierra species. So this is the entire field station in the 1940s, with our iconic Mount Morrison just in the background here. SNARL became part of the UC Natural Reserve System in 1973. And if you're doing your math, that means that we just celebrated our 50th anniversary here at SNARL. Here we are 80 years later. This is our site. Since the 1973 time period, SNARL has grown to become one of the largest and most productive field stations in the entire UC Natural Reserve System. For those of you familiar with Mammoth, SNARL lies on Convict Creek, shown below here, that creek running through, and just below Convict Lake, very near to the Mammoth Yosemite Airport seen at the top here. So at SNARL, we like to say the Sierra Nevada mountains are our backyard and the Great Basin is our front yard. And we think of Snarl as a high elevation base camp for researchers working in both of these environments. Our summer season at Snarl is typically really busy with university classes and researchers. However, we are open year-round at Snarl as our main base of operations here to support research. So we do quite a bit of moving of snow each winter. Just like Valentine Camp, every day in May and early June, we have a busload of kids that comes to Snarl for grade-specific lessons. These range from natural history of bird, trees, or ants to activities in archaeology and Native American life. Programs also combine science and art, and unlike places near UCSB and Santa Barbara, these counties, Moneo and Inyo County, are very remote, and so they're pretty far from a lot of science centers for these kids. All of our programs are supported by donations to the reserve, and we really rely on this support to keep these programs going. Another one of our signature outreach programs at Snarl is our seminar series that happens every Tuesday evening in early May to mid-June. This is at the new Page Center at Snarl. And this program, we host researchers who come and give public talks on topics of scientific interest or local interest. And in a normal year, these are a really great way for our community to come together. We often have food, music, drinks, and everybody has a chance to talk and socialize. So Snarl has facilities on par with a small college campus, including a modern laboratory, housing for classes and researchers, high-speed internet. And then what I like to also point out here is that sustainability is also at the heart of our mission. So we have 7 different solar arrays that meet the majority of our electrical needs. And our newest facility shown in the front here, the Page Center, is a state-of-the-art classroom and meeting space. It's also the first zero net energy facility in the entire UC system. One of our most unique research facilities is a system of 9 experimental stream channels. These were built in the 80s and designed to serve as identical replicates of streams, and they're fed directly from Convict Creek. And researchers have used this system for studies on stream ecology, hydrology, as well as fisheries. Another unique facility is the Snow Science Laboratory, operated in partnership with the U.S. Army CREL, Cold Regions Research and Engineering Lab. For those of you who ski at Mammoth Mountain, you might recognize where that star is. And this facility lies right near the McCoy Station Lodge, and it transmits real-time data and images to UCSB. Snow is a really critical resource for the state of California and forms the majority of the water needs for the state. The research being done here ranges from forecasts of water availability done by mapping snowpack by aircraft to the design of sensing systems for vehicles to navigate in snow and low visibility conditions. So to give you a flavor of some of the animals that inhabit both of these sites, I've just included a few photos from the trail cameras we have around the reserve. Valentine is really known for its bears. The bears here at Valentine utilize the site throughout the season and den on the site in the winter. So they're a very common occurrence at Valentine. Here's another one of our bears, beautiful bear with a white patch on his front, marking this tree. And this year in particular, we've seen a large number of mountain lions on our trail cams at both Snarl and Valentine. So it seemed to be a very good year for mountain lions as well as their prey. Here's one just cruising along the fence line. A lot of the animals around Snarl, the bears and the mountain lions tend to be collared. So they're being followed by researchers at Fish and Wildlife. Here's another bear just cruising along the fence line. So these guys are definitely utilizing the habitat here at these sites. And it's really a treat to have these trail cams and be able to see what's happening at night. So much of the research at Snarl over the years has really focused on the ecology of Sierra Lake ecosystems. And I just want to use this slide to introduce our speaker today, who is really bringing us a new perspective on what goes on in these lake systems. And I borrowed this photo from the Sharon Lab, and I really love this photo because you can tell these guys don't have any fun at all when they're out in the field. You can see John Shuren in the center of the photo. He's a researcher at UC San Diego who has done a lot of his work at Snarl over the years. And Chris Wall on the left, our speaker tonight, is a postdoc in John Shuren's lab. Chris completed his PhD at the University of Hawaii in 2019, and he's been working as a postdoc in the Shuren lab during that time. So he's really switched from marine ecosystems to aquatic ecosystems. He's a physiological ecologist studying symbioses and host-associated microbiomes in both marine and aquatic ecosystems. His research is very interdisciplinary, and it's at the intersection of physiology, microbiology, biogeochemistry, and ecology. And so it's my pleasure tonight to introduce Chris. The title of his talk is Zooplankton Microbiomes Across Alpine Lakes of the Eastern Sierra. And so Chris, I'll let you take it away.
Thank you so much, Carol, for that introduction. It's so nice to hear all the kind words and to just, like you said, see the excitement in everyone's face from that image. We definitely do have a lot of fun in the Shuren lab. Okay, so I'll get right to it. I study zooplankton microbiomes in the Eastern Sierra in the Shuren lab. And I want to just give a shout out to all the colleagues that have helped in this project, undergrad students, graduate students, as well as faculty at other universities. Now, by now, I hope that we all understand that we are truly holobionts or meta-organisms. And by meta-organisms, I mean we are a collection of all of the microbiota that live within us, us being hosts. So in this nice little cartoon here, it shows some trees and grass and plants and animals. And what we see, with our naked eyes, we see these large macroorganisms, but we are really shaped by these microbes, by these very important communities that live within us and on us that can affect our fitness as well as our phenotypes. These microbes can provide us with important resources, metabolites, and importantly, new genetic pathways. And no pun intended, we see this across the tree of life, starting with plants all the way through invertebrates, vertebrates, where symbioses with microbes provide hosts, plants and animals with unique pathways to acquire nutrients or to create metabolites or to mount defenses or digest hard to break down materials. And this is really important in host fitness by unlocking these new pathways. Now, the evolutionary biologist Richard Dawkins described this thing called the extended phenotype. And in the extended phenotype framework, hosts, animals, plants, we are all interacting with our environment, and we modify our environment, and we interact with other organisms in that interaction. So this nexus of the phenotype, which is the physical representation of what an organism is a product of both its genetic material or its genotype and the environment with which it lives and the interactions with other organisms. Now, extending that concept to the larger picture of the microbial age, you could say we have this extended genotype now, where microbial communities affect the host phenotype by providing new genes, new gene pathways that can influence host performance, fitness, and our overall phenotype. The host can also alter the microbial communities that are found inside the water column, in the external environment, and inside their own tissues by providing homes and habitats for these microbes to live. So in effect, hosts modify their environment, and evolution for organisms is influenced by these interactions of communities, both small and large. If we think about how that might affect host evolution, we have this figure here from Henry et al., which is quite salient, describing a host genotype in gray here that is exposed to different microbial communities. And, in some cases, in the left-hand side here, you might see that there is no actual change. The host is not modified by the microbes at all. In other cases, a host may be impacted by locally adapted microbial communities that can provide a new direction of evolution and cause these phenotypes to change through time. In other cases, organisms may see that this shape of their distribution, how they look, how large they are, or a trait of the phenotype may become larger and more variable. So different outcomes can occur with these shifts of phenotypes, changes in the variance in the phenotypes, the physical representation of these organisms. And this is important because it opens up new fitness landscapes for these hosts to exploit. And why that's important is because organisms are looking for these niches, these ways that they can interact with their environment to provide place for their offspring, to acquire resources, and to compete inside the ecological framework. Now, if we take a meta approach here and kind of look at this from a community ecology perspective on microbiome assembly, we could say in each of these little circles here, these might be small microbial meta communities or regional community pools. This could be different lakes, different places, different reservoirs that have within them, as you see these little small squiggles here, that are different microbial communities. So all these communities are differing in composition, their connectivity to one another. They may differ in their history as well as the abiotic factors that they experience. And within these larger regional pools, you have the free-living microbes and you have host-associated microbes. And these are all being exchanged from the external pool into these different reservoirs of species. So let's think of it in this A, B, and C here. A being a phytoplankton, micro community or local community, and B here being the zooplankton of a certain species, in this case Daphnia, which is a common plankton in aquatic systems, or C being a copepod. So in all of these different pools, you have microbes that are associating with these different species that may be really unique. And these unique combinations are exchanging microbes through their feces, through contact, through their dead carapaces or bodies, through their feeding. And all this is going to these other regional pools that are being exchanged across space and time. So in this context, you can think of a microbiome of these hosts being local communities that are nested within these larger regional species pools. And all this theory, I promise, has a point, and that's that we're curious about where microbes come from, why they are found with certain species or certain tissues, and how they're distributed across space and time. Now, in the context of zooplankton, in this case, Daphnia, we are curious about these factors that lead to microbiome assembly and persistence. And you might ask, why do I care about Daphnia? But Daphnia is a model system to help us understand these questions, and it is an important consumer of algae and aquatic food webs. Now, some of the factors that can influence microbiomes might be these deterministic or niche-based effects. These might be environmental conditions, the evolutionary history of the organism, the genotypes that we're examining in different places. It could be their life stage, are they young or old, and what they eat, basically their diet. Other factors that are important are these random effects, including dispersal or ecological drift, or how communities change through time because of random effects. Priority effects are also important, such as what microbes get there first, how they persist, and how they alter succession through time and through the organism's life. Now, this is a great figure here from Akbar et al., showing you some of these different reservoirs, such as the microbes that could be associated with the food that are being eaten by Daphnia, the microbes that are externally living just out there in the water column, those that might be associated with fecal pellets or detritus and small bits of excreted materials, those that are living within the gut system or inside organs. Those can be both those symbiotic ones, pathogens, parasites, as well as those that live on the external tissues of the host. And it's also important to consider where these can be transmitted, i.e., in a young juvenile, do you get your microbial communities from the environment? Are you inheriting them from the place where you exist? Or do you get them from vertical transmission, meaning that you get them from your parent, from your mother, and it's inherited from the mother to the eggs and then goes on to be in the young? So these are all really important questions about where microbes come from, and they extend both from small invertebrates like Daphnia all the way to larger animals as well. Now, other important concepts to understand as we think about microbiome assembly are things like the phylosymbiosis, which is a microbiome composition that reflects the relatedness of the host. And in this image here of a plankton toe showing you many different plankton species, all enumerated here from 1 to 9, This is a phylogenetic tree on the right-hand side that basically could ask, are these species very similar based on their taxonomy, therefore their microbiome would be quite similar, or are they very distantly related, in this case having very distant relatedness of the host and very different microbial communities. So this theory of phylosymbiosis is important because it helps us understand possible mechanisms for how certain organisms have similar microbiomes or very different microbiomes. And lastly here, another important principle is what's called distance decay, which describes how microbial communities can be different across space and time. And this relates to a increase of distance kind of across this axis here. So you have things that are close together versus further away, and how they become less similar as they go further away from each other. This figure here from Martini et al. Shows bacterial communities found within marshes. And they examined these communities within a marsh and asked how different they were from each other based on space, whether they were different across regional scales or across continental scales. And what you see is that there are different patterns kind of shown by these different trend lines here about how similar communities are when they're close together and how different they are when they get further apart. Extending that concept to the Eastern Sierra, this image here I took from the top in Yosemite looking down towards Gaylord and Granite Lakes. But you could ask, are the communities that are found inside Granite Lake here and Gaylord Lake here, are they similar to each other based on their space, similar environmental conditions, possible dispersal and connectivity? Are they more similar than a lake that's further away, somewhere over here, further out into Yosemite? Again, distance is important because it tells us about how dispersal and some of these random effects may be affecting communities, as well as the deterministic effects like environmental conditions and history. Now, if we scale some of these questions and some of this theory from host microbe interactions at small scales to ecological and evolutionary scales, we'll use yet another great schematic from Akbar. And in this case, you have a Daphnia host and you have their gut microbiome that is composed of different bacteria. And then you have a pressure, like a spatial temporal change that affects this microbial community. Maybe it's a dysbiosis or a stressor that breaks down the cooperative interactions between these hosts and symbionts. And you can have two different outcomes here, where one outcome where it's stable, the microbiome performs well, it helps to support host fitness. And despite those challenges, the host performs, experiences increased growth and survival. And maybe you have an ecological outcome of good water quality, high ecosystem services, and lots of fish biomass because of lots of productivity. On the inverse of that, the altered gut microbiome harms the fitness of these organisms. It reduces their ecological role. It makes it harder for them to control algal blooms and produce food for fish. And ultimately, that gets a reduced survival and growth, which may see high algal blooms, reduced ecosystem services. And it's really easy to see which one of these lakes you might want to jump into and gargle some water. I would not do it at the bottom. And I won't name the lake. I don't want to have anyone upset at me on this one, but it's near Snarl South. Now, some of these questions that we might ask in this study that I'm going to be presenting to you are, where do host microbes come from? These associated microbial communities, are they coming from external environments? Are they coming from host selection? And how might we understand these principles and see where these microbes are persisting from? Other important questions are like, what are the factors or the spatial and environmental conditions that lead to stability and changes in microbiomes? Now, I'll investigate that in two principles, one using distance decay and correlations with site-driven environmental conditions to try to answer this question for us. Lastly, are microbiomes similar among related organisms or those with similar ecological roles? This would support that theory of phylosymbiosis and host taxonomic influence on microbiome composition. So we'll go through these and I'll provide some different lines of evidence from our work in the Eastern Sierra, and I hope that you enjoy the ride. All right, so in the Eastern Sierra, we are looking at food webs across different scales. We have really high elevation alpine lakes that are maybe describing this blue food web here. The blue food web is oligotrophic. It has low nutrients overall, but the nutrient quality is high. It has lots of algal biomass, and it is this algal biomass that has all those yummy lipids that are really important for food webs. the omega-3s, all of that. So it's a really high quality food, even if there's not a lot of nutrients in the system. Now, in a green food web, a little lower elevation, maybe montane forest, riparian areas where you have more trees below the tree line, perhaps, you would have high productivity. So you have lots of algae in the water, but it's because there's a lot of nutrients flowing into it from terrestrial sources. So it's this mixture of high quality algae-based food and plant-derived materials that are really hard to break down. So the food quality you might describe as medium because it has both easy to break down, high energy, high desirable products like lipids, and other plants that are really hard to break down and don't decompose well. And the far end of this would be the brown food web, which might be dystrophic in small lakes and ponds. And it's a low food quality, full of terrestrial material, generally looks brown because it's full of these tannins that make the water brown and high dissolved organic carbon. So kind of showing you that elevation difference from a blue, the top here, really clear crystal water, and a dystrophic kind of brown pond here. You can really see visibly the difference between them. Obviously, this relates back to food quality and to the nutrients going into these food webs. We're interested in how zooplankton have adapted to these conditions because despite the fact that these communities are not that diverse, they have the ability to survive across a wide range of environmental conditions. We hypothesize that that ability is linked in some part to their microbial communities. Now, our discipling sites in the Eastern Sierra, we have about 30 of them that we sample, spanning between 2,300 and 3,400 meters of elevation. And you can see the dots here. Some of them are on top of each other. They're really close together. But these are the 30 right here, and kind of going up to 395 right here in this pass. This is Tioga going up into Yosemite, Mono Lake back down here. And across this elevation gradient, one of the biggest things that we see, obviously, is a change in temperature. So those lakes that are low elevation, they have really warm conditions. Those that are higher elevation, much warmer or much cooler conditions overall. And that's what you might expect going up into the mountains, right? Now, visibly, these sites can be quite spectacular. And I have to say, one of my favorite parts of my job, beyond just doing the science, is being in the location that we get to do this science. And it is a true pleasure to be up here and work with these scientists and collaborate with all these people. But these are some of the lakes that are really high elevation. Again, you don't see a lot of trees. It's mostly algal derived food that's in these food webs. And again, it's high elevation, cooler lakes. Go down elevation, you see much more of these trees below the tree line, more terrestrial influences, and the water quality becomes a little bit more decreased as you get especially into areas that have more ponds or dystrophic systems. Now, previous work from our lab has shown that if we looked at the free living environment, just the microbes that are inside the water column, we can see that there are important factors that affect the spatial structure of these microbial communities. Now, on the right-hand side here, this is an ordination analysis. Just know that when you see these plots, the bubbles really indicate samples, and how they separate indicates how similar those communities are to each other. The arrows here show correlations or coefficients of correlation and how similar or how strong the statistical relationships are between the communities that you see and the factors that are on the tips of the arrows. So in this case, one thing that they found in the study was that a big explanatory factor in the free living water communities was whether it was below or above the tree line. And this being above the tree line on this side and below the tree line on this side. Again, think about our figure of that dystrophic pond. Low elevation has more dissolved organic carbon. It's warmer, positive correlations moving with the arrows. It has more of this thing called suva, which is basically watercolor. It's a darker watercolor that indicates lots of this terrestrial material. So what's a big descriptor that helps to explain these communities? Above and below tree line, influence of terrestrial and organic material in the water column. Recent work in our lab has also shown that if you take these fish, trout that you catch in these locations, and you look at their microbial communities, you can see that there's a big difference between what's free living and what's host associated, indicating that these larger organisms show large separation in their microbial communities. And that's really important, potentially because of their ability to foster unique microbes within their gut tract. And it ultimately means that they are selecting different bacteria, keeping it within their system, and they're fostering that to help them perform their functions. Again, trout are not zooplankton. They feed on zooplankton, but very complex relative to these smaller plankton crustaceans. Now, the main focus of my work is to understand more about these microbiomes, or specifically the prokaryotes, bacteria, archaea, and how they associate with these different zooplankton consumers. And here are an array of different zooplankton that we pick out of these plankton toads and we've been studying. They range across sizes here, but broadly we'll I'll describe them to you. You have Holopedium, Daphnia, Polyphemus, Seriadaphnia, Bosmina. These are all cladocerans. Then you have two types of copepods, being calanoids on this side and cyclopoids on this side. So these are two main groups of plankton that we see in these lakes. And one of our big questions is, we're looking at the influence of environment and their geography and host species. We want to know what structures these zooplankton microbiomes in the Eastern Sierras and how it might relate to their performance and evolution. Now, how do we do this awesome work? I know that's on the question everyone's asking themselves. Well, we go out in these lakes. You can see my daughter here, my field research scientist in training here on my kayak, and we're going out to these lakes where we take water samples, we take environmental measurements, we collect plankton inside these large plankton toes, and we bring this back on shore where an army of awesome undergraduates and graduate students work to help prep and sample and preserve these samples, bring them back to the lab, and then we pick through them on the microscope, separating them by species. It's arduous, it's fun, it's the best times, it's the worst of times, but we get some really great data, and it's a pretty fantastic gig. Now, once we have all of those plankton separated, they're enumerated, and we know how many are in each tube that we go through DNA extraction with. So we take those plankton, we extract their DNA, we then run it through a PCR, which is a method for amplifying a specific target of DNA, specifically one that aligns with prokaryotes. So we're interested in just looking at those prokaryote DNA sequences. And we run that through a MiSeq platform, which then will sequence the DNA and tell us all the sequences of the prokaryotes, and then we can align them and figure out who is present, basically. Now, showing you some of our results that we have from this work. This first figure here is a relative abundance plot. And in all these cases, you'll have zooplankton going across the x-axis, and you'll have the furthest one here being water. And I'll try to note that it's water so you can kind of visually make a visual comparison here. But in some of the most dominant bacterial classes that we have here, there are a few that I've outlined. And just to think about some of their functions, the actinobacteria are really important heterotrophic bacteria plankton that live in the water column. The Bacteroideae are those that are important for humic matter digestion, which is like plant material breaking down. The Gamma proteobacteria are really important in nitrogen cycling and respiring nitrogen, which is an important pathway for prokaryotes. And the Verrucomicrobidae, which is a really diverse carbon cycling group. And so with all of these together, we kind of look at the dominant distributions here. And one thing that we see is that we have a pretty unique distribution in water relative to other groups. They don't all look the same. We see a lot of the same classes overall, but for instance, like the actinobacteria, much higher abundance in water, or these Verrucomicrobidae are very more abundant inside waters compared to other samples. So as a coarse look, this is really interesting. And one thing I want to kind of point out here is these blue calanoids, which we see in one specific pond location. They have these really high abundance of these Bacteroideae class bacteria, which is important for humic matter digestion. And you have lots of humic matter in ponds. So it suggests that at least part of this is driven by location. Part of it might be driven by the host taxonomy, but they look similar to water, but they don't match it completely. If we look at the richness, which is the total number of bacterial taxa that we see, so kind of basically counting, enumerating the absolute number of species, bacterial species we'll call them, but they're actually just amplicon variants or amplicon sequences. We could count these up and ask how many species are present. And what we see is overall, we have around 200 different bacterial richness or alpha richness species, however you want to refer to it, found within water. And we have much less diversity across all the other samples of zooplankton. If we take a different sort of take on this and we run it through another metric that's a diversity matrices called the Shannon diversity metric, we can look at that and say, do we see something different when we take into account both richness and the evenness of the distribution of these groups? We do see a similar pattern. We see that once again, no matter how we slice it, there are less bacteria found within zooplankton relative to water. So overall, we see that alpha richness, which is this enumeration of groups, by species shows a lower richness in animal hosts relative to the environment. Taking all these lines together, we can kind of ask that first question again about the sources of host microbiomes and if there is host selection. And it appears that the water might be a source for many of these bacterial taxa, but hosts are fostering unique microbial communities and they're selecting different ones to live within their tissues. If we build on that and we ask, what about these geographical patterns that we might see in richness across these lakes? One thing that we see is we can take the log richness of the total number of bacterial taxa. When we look at it across elevation, we see a trend, although a weak trend, where there is a change, increasing richness with elevation. It's an interesting thing to note here. We don't have a lot of super low elevation sites, but it's interesting to think that the elevation alone might be influencing the amount of diversity we see. Looking at this at the site-specific level, we see lots of variability across these different sites. And while some of this looks like it might just be stochastic noise, one thing that's really interesting to me on this first look is that as we look down this gradient here in low elevation lakes, these are all ones that are very dystrophic or more pond-like. They're still classified as lakes as far as these 1, 2, 3, 4, but they have lots of nutrition flowing into it, lots of riparian zones around it. They're shallow. And this furthest one, most distinct here, is actually the pond. So we see that there might be this influence overall where high nutrients is influencing the amount of richness inside these lakes. And it's really important for us to think about is how these lakes change through time and also how they're influenced by the environment and humans. If we want to step back from that and say, well, what about larger patterns that we might see across distance? We can separate this by looking at different groups of these zooplankton, copepods being both the calanoids and the cyclopoids, which you see just broken down here. And we ask, again, what's that distance-based relationship? Well, this is a little bit different than the one we saw at the beginning, but overall, we see that the distance going across the x-axis here, increasing distance, and through this direction, you have the most dissimilar. So those that are close together show greater similarity than those that are further apart. Maybe not such a crazy thing to think about, but this sort of structure is important for us to understand what is leading to changes in these microbiomes. We see the same pattern when we look at the cladocerans across these 4 or 5 different species from Daphnia, Cereodaphnia, Bosmina, Holopedium, and Polyphemus. We see the same kind of relationship where communities that are close together have much more similar microbial communities than those that are further away. And some of these patterns are quite strong, being much stronger than what we've seen even with some of the trout species. So it's an interesting finding that shows that distance is a really important metric in describing the spatial distribution of these microbial communities and how they change relative to different species, different places. And so for these two lines of evidence together, looking at the richness by elevation, and this is another take on the distance decay relationships, showing all the water samples, all the copepods, cladocerans, or all the samples together, we see that across all these samples, there's a trend for increasing similarity when you're close by and an increase of richness with elevation. So we conclude here that geography and distance appear to be key determinants in these communities, and they're shaping what is found where. And we see this conserved across different sample types like water and really strong signals with these zooplankton hosts. Now, we'll run this through a distance matrix or a distance-based matrix here. And this is another ordination plot where you can look at these ellipses, and big takeaway here is each one of these points represents a sample. And that sample is, 50 some odd individuals of a certain species from a certain lake. And if we look at just the taxonomic identity, genus, or the functional groups being all calanoids or all cyclopoids, we can kind of see how these separate in space. I gave some arrows here to help you see where these all fall because they're sort of on top of each other. But what's really interesting is that water is highly conserved, all nested here in the middle. You have different communities that sort of branch out from that. And in some cases, they're quite similar to water, like Holopedium up here or the Polyphemus down here. But as we move out to the sides of this ordination, you see other important patterns like Bosmina and Daphnia and Cereodaphnia, all cladocerans, showing nestedness on this side of the ordination plot, whereas calanoids, these other copepods, are falling across on this side. So you see that there's a pattern here where these species are separating. They're unique relative to the water column, and they show to what I view as a similarity across taxa, being that you have more of one taxa on this side the cladocerans, more of the other taxa on this side, the copepods. Very interesting finding, and I think it really tells us a lot about what's driving some of these factors. If we want to look at that one more way, we could run a different type of ordination, again, taking those communities, blasting the shotgun of Skittles against the computer screen, and looking at this in a rank-based non-metric ordination, meaning that this is going to order them in a certain way. It runs just a different ordination. It's all about an algorithm. Don't worry too much about it for the talk. But what I want you to take away from this is that, again, the communities separate. You have Daphnia on this side. You have calanoids on this side. You have different groups across the matrix here. Water, again, being unique and not interspersed across the samples. Other things that are happening here, these vectors, the strength of the correlation with the axis being NMDS1, the X, or NMDS2, the Y, the strength of that correlation indicates by the line of these arrows. And one thing that we see is that you have, again, the calanoids are positively associating with locations that have high DOC, high phosphorus, and higher temperature. So what is one key factor that might be shaping these relationships? It appears to be environmentally driven and taxonomically specific. So I think this is really exciting because it talks about, again, this evolution that's happening at the sites where these plankton are, and how despite being in different places, you have nesting of the organisms. So it talks about evolutionary history of the hosts and the role of the environment shaping their microbiomes. And we've done some tests to try to understand more about what is driving changes, or can we cause changes in host microbiomes. In one study, we did a combination of adding lots of plant material and different kinds of plant material, those that were exposed to fire or those that were not exposed to fire. And through these, you can kind of see a comparison of how we can make the water super dark with lots of plant material, or not so dark with less plant material, and how this affects water column chemistry, being this giant increase of dissolved organic carbon across the material loading gradient. And so we asked, after 89 some days, when the conditions had subsided, those organisms inside the community, specifically being Daphnia or mosquito larvae, how they compared to the microbiomes of leaf material that was inside the tanks or the water column. And again, one thing we see is that there are unique reservoirs of microbial communities. The water looks unique relative to the detritus that's in the tank. Mosquitoes kind of bridge the two, maybe because they're feeding more on the detritus at the bottom of the tank, and the Daphnia maybe are using more of the water column productivity from algae. So maybe there's feeding differences that are driving this, but also, again, taxonomic influence. And do the Daphnia here look more similar to the water or to other conditions? Could we drive a change? Broadly speaking, it looks like the Daphnia were really conserved. They were doing something that was unique to the conditions that were defined by their environmental history, but also their evolutionary history. And I think that that's a really interesting finding. We have looked at this in other ways in Yosemite as well, about wondering how these trends across time and space may affect the water column. And so we've sampled across 2016, 17, 19, and 21, and we have data from 2022 we haven't incorporated here. But broadly what we see here in the water column is that across these times, you have a significant site variation, i.e. these dots don't always fall in the exact same place. But overall, the ellipses that you see drawn here are right on top of each other, suggesting that the sites may vary, but across the years, there's actually a very stable environment inside the water column. So we're still understanding more about what is driving these factors in the water. And I think that what's powerful about this is it just describes across different years, different conditions, we see a conserved relationship with the microbiomes in the water, the free living microbial community across many different lakes in different places and times. Now, some big takeaways here that I'd like you to sort of walk away from this talk. A little bit about microbial theory, although I understand if you Control-Alt-Delete that as soon as you walk out of this Zoom meeting. But broadly speaking, what we find in these beautiful, unique settings that are just some of the most beautiful places on Earth, we see that these communities of zooplankton, they foster unique microbial populations that are different than the water column. We're still understanding what are the drivers of those changes, what drives the zooplankton community to be different from the water, but we see that there is a pattern of selection. So the zooplankton appear to be selecting certain organisms to live within them, or fostering environments that allow those microbial populations to live within and on them. And the hosts, either through evolutionary pressures or through other feeding or other activity behavior, are appearing to drive this conserve microbial community structure. We do see there is a distance decay relationship and that environmental conditions are structuring host microbiomes. And we saw that pretty clearly with, like over here with the copepods, showing that increase of similarity when you're close by and a decrease of similarity when you're far away. And we saw that pretty clearly with the other ordination plot, showing which environmental conditions were positively associating or negatively associating with different microbiomes of different host species. And finally, we see that phylosymbiosis, this shared history or shared environmental relationship, might be driving some of these similarities in microbiomes for different zooplankton. And that's really interesting because I think it talks to the reservoirs that live within tissues, different gut tracts, different communities that can be fostered within host species. And I think that speaks to the evolutionary pressures that shape those phenotypes and ultimately their evolution through time. And that's what we're looking at today. And so with that, I'm very excited to share this with you all. A lot of this is completely new data, so forgive if it seems rough and if I'm too excited about it. So I hope that excitement carries over to peer review. And I want to thank all my collaborators on this project, and really have to just say thank you to Carol and the Snarl community for always hosting us, taking care of us, and all the awesome undergraduate researchers that really made this research possible. We couldn't have done the field work, we couldn't have done the lab work without you. And so just big thanks to all of you. And with that, I'll take any questions if you have any.
All right. Thanks so much, Chris. One thing I want to just say is how nice it is to see the results of all your work, because I get to see you guys all summer long, busy out working, and it's so fun to hear about all the exciting stuff that you guys have found. So I just want to remind everybody out there, if you have questions, you can type them into the Q&A area. We have a couple of questions coming in on the chat, but I'll start off here. First off, with John Shuren's got a question. John is asking, do you see any signs of human impact in the microbiomes of remote alpine lakes?
That's a great question, boss. On first pass, I wouldn't say that I do. And it might be that the lakes that we're looking at are pretty pristine. We've talked about other locations that could be more human impacted or more disturbed. I know that some of these lakes do not have fish, or have now been fishless. There's at least one or two of those locations. But broadly, I think that we might see some of those patterns if we look for specific taxa, or we started to mine the data to look for taxa that might be more indicative of human influence, specifically maybe it's cyanobacteria or other groups that might be found as pathogens. And I think it's really an important question because these lakes are frequented by humans often. They're definitely going to be impacted by climate change, being high alpine locations. So it's a very interesting question. I look forward to working on that with you.
All right. Got a question here. Next question is from Scott Cooper. How much is known about the genetic structure of the host zooplankton populations? Does spatial autocorrelation in the microbial communities reflect spatial autocorrelation in zooplankton genotypic variation? And do different zooplankton genotypes within a species have different microbial communities? So that's in the, you can read that, Chris. I know it's a three-part question there in the Q&A if you want to.
Yeah, I mean, that's a fantastic question. I think that one thing that we tried to build on in this work was that there is a wealth of studies in the literature that take a single genotype and breed it for 10 years in the lab, and then they run a microbiome experiment on it. Or they might do 3 different genotypes and to compare genotype effects. And what is very troubling or exciting, depending on how you look at it, is that genotype matters a lot. And so, in these lakes, I don't know the exact genotypic structure or how connected some of them might be in that regard. Daphnia, as you probably know, are predominantly parthenogenic. So there's a lot of that going on in every single lake. I think it's something we could mine with the data that we have, but we took this as a population approach for each individual lake. And I think it's something that we will have to consider but I don't know the answer for what the structure is like across these lakes.
Okay. Next question is from Paige Hiller Adams, asking about the benefits the benefits to the hosts of their microbiomes. I'm just reading in the chat here too, suggesting maybe experiments with cleanses, things like that, survival. So essentially, like, what role do these gut microbiomes play in the sort of health and fitness to these zooplankton?
I think it's a very exciting line of research, and there are studies that have taken the more the hands-on manipulative approaches to addressing microbiome performance and fitness. Of course, you can take other avenues by doing like metagenome analysis to actually understand more about the function of these communities, as opposed to just characterizing taxonomy, who is there. So I think that going forward, one thing that John and I are looking forward to doing is exploring those questions by looking at metagenomes as one avenue to approach that. But secondly, we did do a follow-up experiment that I didn't have time to talk about today, where we modified conditions inside a bunch of different mesocosms at Snarl. And through these modifications of being high plant-derived nutrition or very low nutrition, like really low nutrients, how those conditions would alter the microbiomes, and then did a reciprocal transplant between all the conditions to ask if you had different Daphnia from different places, effectively putting them to new environments, would that lead to microbiome change, and what that might mean for fecundity, egg size, actual phenotype size, and other traits for fitness. So it's one thing that we really want to do more of. Although, of course, in the case of being at Snarl, it's hard to stay on site. We want to go out and hike and go do some field work. So it's another avenue that we're exploring, and it's a great question. Thank you for asking it.
Okay, I don't know if there's any more questions. Let me ask, oh, wait, there's one here from Doug Bush. Are there measures of stability of the microbiome, and do those stability measures change with elevation or nutrient loading of the water body?
It's a great point. I think one thing that we have tried to address in a follow-up study was, if you went to the same lake and sampled it many times, would you see the same kind of snapshot, or how much change would you see if you tracked it through a season? Some recent work in other locations, not in the Eastern Sierra, but they looked at how the microbiome of organisms, of Daphnia specifically, tracks nutritional and temperature changes. So going through a season, you would see that the microbes may be changing the water column as it goes into spring and into summer and finally into winter. And that change in the temperature that affects productivity, turnover, and the microbes that are exploiting those nutrient landscapes, feedback to influence the host microbiome. So there are studies that are kind of looking at that, and I think that we might be able to mine that with our data, because we have it across several years and several different places as well. So it's a great thing that I'm excited to look into more about, understanding what leads to stability, what might be drivers of change, and how these microbes all kind of come together in different networks that are based on space or on host taxonomy.
And if I could, Chris, could I ask one final question of you? One of the things is, here at Snarl, a lot of researchers have been focused on, over the last many years, are looking at differences between lakes that contain fish and those that do not contain fish. And I was just curious to see if you have samples from fish and fishless lakes, and if you're seeing any significant differences due to the fish presence.
We do have one lake that we sampled that does not have fish. I don't know if it has always been fishless. There might be others that are really, I kind of go by our colleague Josh Dominguez. If he can't catch a fish, it might be fishless. So there is actual metadata on this, but we purposefully avoided those historically fishless lakes because of the possibility that our kayak and our sampling team might inadvertently add or introduce the fungal pathogens that are really deadly to the native yellow belly frogs. So it's a big concern for access. And if I could build on this research, I might ask the frog team to go out and see if they can do a plankton tow and preserve. But one thing that we were really curious about was how these communities would be, if they would work, if they could be preserved the way we were preserving them, taking them back, putting them in ethanol, if we would get reeds out of them. Luckily, Snarl has a -80. And so as long as we would go out for the day and come back, we could freeze it and guarantee that it would be safe and preserved. But more remote locations might require more delicate or experimental ways of preserving the DNA of the hosts and the microbes.
Yep. Okay, Andy, do we have time for one last question?
Yeah, go ahead and take the last question about plastic pollution, and then we'll wrap things up.
Okay, sounds good. So the final question is from John Ritter, asking about has plastic pollution made it into these alpine lakes and into fish and other animals?
Great question. I think that if anyone's been to the highly fished areas, you might suspect that the answer is yes, based on the amount of nylon thread you see. I would say that, broadly speaking, I didn't catch any fish. I wasn't focused on the fish, right? But I know that we do see lots of human traffic, especially with fishing and lines and hooks and things like that that are around some of these lakes. It's pretty special when you get away from all that and you don't see this. Good anglers packing up their mess with them. But I think that most of the lakes we go to, despite being pretty close by major trails, there's really not that many people that seem to be there. So we're really fortunate to go to places that are easy for us to access overall. 10-mile round trip hikes, but we get to pretty fantastic backcountry areas that are quite pristine. And we're really thankful to have that opportunity.
All right. Well, I want to thank you, Chris, so much for your talk tonight. It was super interesting.
Thank you.
Andy, any final words?
Yeah, I want to thank both Chris and Carol for some great presentations tonight. And just remind people there are 2 more seminars to go. Next week, we'll be talking about snowy plovers at Coal Oil Point. And the last seminar will be 2 weeks from today. We'll be talking about bats on Santa Cruz Island. So make sure that you join us the next 2 Wednesdays at 7. Several people have asked whether or not this webinar has been recorded, and it has. And so I will upload the recording to the UCSB NRS YouTube channel sometime tomorrow or Friday. So just call up YouTube on your favorite web browser, type in UCSB NRS into the search box, and we should be the first thing that pops up. I'm going to sign off right now for everyone, but I'm going to put a screen up just for a couple minutes that have a couple web addresses if you'd like more information about the UC NRS in general, the UCSB NRS, or if you're interested in trying to scribble out the actual UCSB NRS YouTube channel address. It's sort of long. So with that, I'll say good night, and I hope to see you all again next Wednesday.
Thank you all.